Okay, it's 9 o'clock sharply. I think we can start the event. Ladies and gentlemen, I would like to welcome you all to this technical meeting on FEEDAP panel guidance on the renewal of the authorization of feed additives. It's always a great pleasure to meet you in such events organized by EFSA and the feed unit. I would like to thank all organizers for this event. It's well known that regulation number 1831 of 2003 establishes that feed additive authorizations are valid for 10 years and also renewable for a 10-year period time. The procedure for renewal is described in Article 14 of this regulation. An application for renewal has to be sent to the Commission at least 1 year before the expiry date of the authorization, and the technical dossier is to be submitted to EFSA for the assessment. The FEEDAP panel consistently updates the relevant guidance documents for preparatory work required for the feed additives application. Thus, during the last years, the FEEDAP panel has adopted a series of guidance documents to help applicants prepare and submit technical dossiers for the authorization or renewal of the authorization of feed additives for use in animal nutrition. Specifically, there were 3 guidance documents adopted in 2017, the one for the safety of feed additives for target species, for the consumer, and for identity and characterization of the conditions of use of feed additives. In 2018, another 2 guidance documents were adopted for the characterization of microorganisms used as feed additives or as production organisms, and for the efficacy of feed additives. Another one was adopted in 2019 for the safety of feed additives for the environment. The FEEDAP panel has recently updated the guidance on the renewal of the authorization of feed additives. The old one existed since 2013. This updated document was adopted on November 2020 and entered into force on 27 of March 2021, just a couple of weeks before, and covers all aspects from the characterization of the additives, the safety for the target species, consumers, users, and the environment, and of course for efficacy if applicable. The objectives of today's meeting which is a web-based technical meeting with all of you, the stakeholders, on this updated guidance document, is in order that EFSA has the setting and the opportunity to explain the current scientific criteria for the assessment of the applications for the renewal of the authorization of feed additives, and also to discuss questions posed from you and exchange views on scientific issues related to the preparation and the risk assessment of applications. Now, before I give the floor to the colleagues from EFSA, I would like to make clear that the current meeting will not cover the administrative process of submitting a renewal application, nor the corresponding provision of pre-submission advice, or other activities related to the implementation of Regulation 1381 of 2019 on the transparency and sustainability of the EFSA risk assessment in the food chain. At this point, I would like to give the floor to Rosella Brosi, who is a scientific officer in the feed unit, to present a brief introduction and some in-house messages. Rosella, you have the floor.
Thank you, Vasilis. Good morning, everybody. Welcome also from my side. My name is Rosella Brozzi, and I'm a scientific officer of the feed unit. As Vasilis said, we will start with some tips, technical tips. As announced on the web, this meeting will be recorded, and the recording will be made publicly available on the website. You have granted your consent for this during the registration phase, but if you change your mind, please exit from the meeting now. Please keep your microphone and camera off during the whole meeting unless specifically asked by the chair. Please refrain from writing on the chat, except for posing questions or requesting the floor. And if you face any technical issue, exit and rejoin the meeting, please. But if you still have problems and they persist, you can write to the email address that you see on the screen now. Margarita can give you some technical support to sort out these problems. We recommend you to use the headset to guarantee a good quality of the audio. Now some organizational details. To express interest, please use the chat box where you can write your questions, comments, or you can request the floor. To maximize the plurality of interventions, you will be given the floor just once. You might be given the chance for a second intervention if time allows. We hope that the PowerPoint presentation that my colleagues will shortly show trigger many questions. So to facilitate the organization of the replies, please post your questions, follow the order of the topics tackled in the presentation. At the end of the presentation, we will have a session dedicated to questions and answers. We will start by addressing the questions received during the registration phase. Then we will switch to the questions triggered by the presentation, organized by section, as I said. And within each section, we will try to reply to the questions in the order as received. At the end of the section, and each section, and at the end of the Q&A session, we will open the floor for any additional questions. You will be able to pose questions orally once the chair has granted you the floor. When taking the floor, please activate your microphone, and if you wish so and your network allows, also the camera. When you take the floor, please do state your name and affiliation. For the benefit of time, thanks for being as brief and clear as possible. And with this, I thank you for your attention. I wish you a good meeting, and I give back the floor to the chair.
Okay, thank you, Rosella. Now, I would like to give the floor to Jaume Galopart and Matteo Innocenti, who are scientific officers in FEED unit, to present this FEEDAP guidance on the renewal of the authorization of FEED additives, and also present novelties and changes regarding to the old one guidance. Jaume, you have the floor.
Yes, thank you, Vasilis. Good morning, everyone. My name is Jaume Galopart. I'm also a scientific officer in the FEED unit. And today, together with Matteo, we'll try to give a little bit an overview of the main novelties on the guidance on the renewal of the authorization. Let's briefly start with some of the history that the chair has already given a very brief overview before on the guidance on the renewal. The first guidance was actually adopted by the panel in 2013, and that was a very brief guidance following the requirements of the regulation 429 at that time. It was done in a time in which I think we had only received 1 or 2 applications for the renewal, so the experience of the panel on the renewal applications was rather limited. In 2016, the panel did some work in revising the need to update the different guidance documents. And as an outcome of that, it came clear that most of the guidance of the panel needed to be updated. And this is what the panel did during the period 2017-2019, in which the vast majority of the guidance documents for the assessment of FEED additives were updated. The last one in this process, it's the current one, the guidance on the renewal, which takes into account the experience gained by the panel during the assessment of the many applications for the renewal that we have received in the last years, and also all the scientific knowledge that was included as well in the updated guidance that you have on the screen. This guidance on the renewal entered, let's say the implementation date was 27th of March at the same time as the transparency regulation. So that means that all the applications for the renewal of authorizations of FEED additives submitted after this date should follow the requirements listed in this guidance document. So first thing is that it should be clear that renewal is not a second re-evaluation. The first assessment was already done at the beginning when the original application was sent. So with the renewal, we do not expect that as our extensive assessment is done as it was done for the re-evaluation. The objectives of the renewal are quite clear, are mainly two. The first one is to demonstrate that the additive that is placed in the market fulfills the conditions of the existing regulation. And the second, that to demonstrate that in the light of the current knowledge, the additive remains safe under the approved conditions of use. So what are the approved conditions of use? To start with, it's the additive composition. We need to make clear that what we have in the market is exactly the same as it was authorized. And that means the composition in terms of active substance, any potential impurities, method of production if listed, the target species or the conditions of use for which a species it is authorized, if there is a minimum or a maximum. But let's not forget also the other provisions which in cases contain also some limitations or other conditions for the use. We are aware that applicants take the chance when they submit an application for the renewal to also send applications for other uses, for example, an extension of use to a new target species, or maybe some modifications on the authorization, increase or decrease the maximum use level, changes in maybe in the composition or in the manufacturing process. All these joint applications or renewal plus a new use are perfectly fine. It's possible to do them, no problem. But this needs to be reflected in the application that is submitted to the European Commission. And second, all the relevant data to assess these other, let's say, applications should be submitted in the dossier following the requirements listed in the applicable guidance. So, for example, if you claim a new use in a new target species for which the additive was not authorized, then the data on the safety and efficacy should follow the guidance in force for this respect. So now let's go into the details of the guidance. We will start with the section on the identity and characterization. So in order to ensure that the additive that is in the market is actually complying with the conditions of the authorization, it's important that the additive is fully characterized. So for that, we need that the dossier contains a complete section too. All the data necessary for the characterization should be submitted in the dossier. But that does not mean that you need to provide actually completely new data for all the different sections of the dossier. It's possible that you can, let's say, recycle some of the data that you originally submitted for the dossier 10 years ago. And let's see now in the next slide, what do we mean by that? So in this table, you have the main sections of the characterization or the main subsections. And on the right column, you have the requirements for new data. Those are in red. When there is a yes, and in red, it means that new data needs to be submitted for the applications for the renewal. If there are no changes in the composition or the manufacturing process of the additive, the need to provide new data is rather limited to the quantitative and qualitative composition of the additive, what we know as batch-to-batch variation. In that case, there is the need to provide data from 5 batches. That's new. In the past, it was only 3. Now the requirements of the guidance is data from 5 batches produced during the last year, and 3 batches for the impurities. The rest of the section, basically, you can submit more or less. There is no need to submit new data. You can probably recycle most of the data from the original application, with the exception of the characterization for microorganisms used as additive or as production strain that we will see in a minute. For the other sections, only if there is new data that the applicant considers relevant, this should be submitted. But what happens if there have been some changes in the composition or the manufacturing process or any other aspect in the production of the additive? In that case, there is the need to provide information on these changes. So there is the need to provide probably an update on the manufacturing process if there has been a change in the manufacturing or the composition. And then, depending on the change, data on the physical state of the additive or, for example, stability and homogeneity might be needed. Let's give an example. If you produce a product now, you change the manufacturing process, and this means that the product you are producing now has a different particle size distribution and a different dusting potential. So it's likely that you will have to provide additional data on the dusting potential. And then this should also have probably to be considered for the assessment of user safety. And also, if the composition changes, that may have an impact on the stability of the product, that should be properly addressed by submitting new data on the stability. So what happens with microorganisms? I said before that for microbial products and also for fermentation products, there is the need to provide basically in all cases additional information. And this is why the characterization of microorganisms has been the subject of this new guidance that was adopted a couple of years ago, in which several new requirements were introduced. So we ask you to have a look at the guidance and to ensure that the requirements for the identity and the characterization of the microorganisms follows the requirements of this guidance. What has changed basically is that there is the requirement, for example, of the identity for bacteria and yeast to be identified using whole genome sequence. And we have seen some cases in which the taxonomical identification has been updated. Also, in some cases, the list of antimicrobial substances for which you need to test the susceptibility has changed, or the breakout cutoff values have been changed. The same for the toxigenic and pathogenicity. So all these aspects need to be considered and updated according to the requirements of this guidance. The only exception might be the case of production of antimicrobial substances, that if this has been demonstrated in the past, there is no need to redo the test again during the renewal. Last, it's the conditions of use. That's quite important that you pay attention to this. So the additive is already in the market. There are conditions of use for which the product is authorized. What we need to know in the application for the renewal is whether or not the applicant proposes to keep the same conditions of use. If the conditions are the same, this probably will mean that some data is not necessary for the efficacy or for the safety. But if the applicant proposes to modify the conditions of use, for example, the reduction of the minimum content or an increase of the maximum content, then the implications for the safety and efficacy need to be assessed. It's quite probable that if you request to reduce the maximum or to increase, the maximum authorized content of the additive, new data on target animal safety or maybe consumer or even the environment might be needed. On the other side, if you propose to reduce the minimum content of the additive in feed, maybe new data on the efficacy needs to be provided. It's also important to consider that in the authorization, in many cases, there are other provisions that include some relevant data. It could be, for example, that there are the concurrent use with coccidiostats. If the applicant wishes to include more coccidiostats for which it is simultaneous use is requested. So also this should be taken into account. Just to close the section on the characterization, in this slide, we try to summarize some of the issues that we have seen that applicants have some difficulties to comply with the requirements of the previous guidance, but also with the current one. The first one is the data on the composition and on the purity. We have seen many cases in which there is no new data, that applicants submit the old data from 10 years ago, or that so that the batches are older than 1 year, maybe data from 5 years ago, or that not enough batches are analyzed. In some cases, there is no clear indication whether the applicant has introduced any changes in the manufacturing process or the composition. All this should be clearly stated in the application, in the dossier. I think one of the most common is that the characterization of the microorganisms used as the additive or as production strains are not really following the requirements of the new guidance. So there is no identification using the whole genome sequence or for the antimicrobial resistance, the same thing. Or quite common also that there is no up-to-date data on the absence of viable cells or DNA in the final products. Also, we have seen that sometimes the additive is authorized under certain conditions and the applicant proposes in the renewal different conditions without specifying that this is a new request. So there is not clear understanding whether or not the applicant proposes to keep the same conditions or to propose new conditions of use. And last but not least, when there are these joint applications, for example, with extension of use, sometimes there is quite a confusion for which target species the applicant intends to request a new authorization. So we ask you to please follow quite in detail the guidance and to pay attention to all these details when submitting your application. So with that, we close the section on the characterization, and now I give the floor to my colleague Matteo Innocenti. He will continue with the rest of the presentation.
Yeah, thank you very much, Jaume. Good morning, everyone. I'm Matteo Innocenti. I'm also scientific officer in the feed unit. And today I will take care of explaining a little bit the main characteristic, the main feature of this new guidance regarding the safety, and a few words also on efficacy of products submitted for the renewal. I just take the chance now to recall what was briefly explained in the beginning. Regarding questions, if you have questions, you can put it in the chat. You will see that the safety part is divided in different subsections, more horizontal one at the beginning and then specific for each of the different safety sections. So if you have questions related to each of these parts, you can put it in the chat when we will be presenting the different parts, so it will be easier also for you to follow. So having said so, we can move a little bit. So safety. As my colleague Jaume said at the very beginning, the whole scope of the renewal is to demonstrate that the additive that is on the market remains safe, and remains safe for the target species, the consumer, the user, and the environment. So this is the most important things to take in mind when preparing an application for the renewal of the authorization of an additive. So to demonstrate this, an update of the information on the safety would be needed. An update means that since the safety was originally demonstrated when the additive was firstly evaluated and then authorized, a time span that covers since the last authorization or even the last renewal and 1 year before the submission, just to give you the time to prepare all the necessary information for the submission, would be needed. A certain amount of information would be required to assess whether the additive remains safe or not. Some of them could be sent if they are available. For example, the quantitative data on production or on the use of the feed additive and the geographical distribution. This is something that will give a picture, will help to have a full picture of the use of the additive, and therefore just to put into a context the evaluation of all the other information that would be then submitted. You may imagine that an additive which is produced and distributed in very few, in a very small amount and in a few regions, might trigger the production of fewer information regarding the safety, and instead additive that are used in a very large amount all around the world could lead to a higher amount of information. Which are these information that instead would be really always needed to be submitted? If it was requested by the authorization, the post-marketing monitoring plan, so the outcome of the post-marketing monitoring plan, should be always submitted in the dossier. then any possible report of any adverse effect which has been made available to the producer or the user of the additive during the period in which the additive remains on the market authorized. These are the two main set of information that would be always needed. And then a set of additional information, additional evidence would be needed to support the safety. How this additional evidence could be retrieved and then made available to the panel for the evaluation. This is the major, we can say, novelty of this guidance compared to the previous one, because the panel considered to go a little bit more in detail on how to submit this information and mainly how to retrieve this information. So the first possibility that the applicant has is to do and then to submit the results of an extensive literature search. The extensive literature search, so it's an instrument that could be used to provide the information on the safety under the currently authorized condition of use, so in the period in which it was authorized and on the market. The extensive literature search might respect some minimum characteristics that were defined and described in the guidance. And these main characteristics are that should cover the period that we have said before, so since the last assessment until 1 year before the date of the submission, should allow the full characterization of the additive. It means that the information retrieved should be related to the additive under application. If the additive is not identical, the information should anyway allow a conclusion for the additive under application to be made, for example, with the read across, and the reasons to support the read across should be always explained in this case. That means that to fully characterize, so to be sure that the additive is exactly the same, we have two more and additional specification to be done. So for those additives that are produced by fermentation, the production strain would be needed always, so we are sure that we are talking about the same microorganism. And in case of mixtures, complex mixture, all the different components of the mixture should be looked for, so that means the individual components, and then also the complete mixture should be taken into account. So, as I said, in the guidance, there is a thorough description on how the extensive literature search should be conducted. And in the guidance, there is also the reference to the publication edited by EFSA, which could guide you and help you in performing the literature search. But then the panel decided also to give us some characteristics directly in the guidance to help you and to guide you in performing the search. I will go briefly through some of them, those that are considered the most relevant ones that are described in the guidance. So a few aspects that should always be considered. The first aspect to be considered is where to look for the information. Since the idea is to demonstrate that the additive is still safe, remains safe, so the information could come from a series of different sources, not only the classical journal or reports of peer-reviewed paper or books, but also any other source of information like a website of conferences, website of organization, a reference list or of other journals, and so on. So in order to have the larger bucket of information as possible. And to be sure to catch all the relevant information, also importance should be given to the search terms and the search strategy which is applying in doing the extensive literature search. So a lot of attention should be posed in the search strings, the effects that should be followed or looked for, to be sure to identify the additive properly, for example, with CAS numbers, Flavis numbers, the generic name or the brand name of the additive, which is important, and also to use all the different operators which would allow you to have a complete search. So it's not only important how the search is done, but also how it is reported, because this will then allow the panel to perform the assessment and to evaluate the information, the data provided. So also for the reporting, few details are included in the guidance. In particular, attention should be posed in the main characteristic of the search, like the databases in which the search was done, when referring to databases, the date of the search and the date range, so the time span that we have said before, any limit, the full search strategy, and so on, in order to make the search also repeatable and to give the possibility to evaluate the results. When instead reporting the search is done in website or journal table of contents or any other source, is important to make reference to the name of the source or where it could be found, also in this case the dates, how the search was done, any limit applied to the search, and so on. When looking for reference list, instead, it's important to report all the bibliographic details, obviously, and also the number of the references that were retrieved in this extensive literature search, and then any criteria applied for the selection, so inclusion, exclusion criteria, which led to the selection of the information of the paper that then will be reported. This will be the weight of the number of papers that were excluded compared to the ones that were retrieved in the search. And it is important that the reference for all the relevant publications are reported in the dossier. This is regarding technically how to do and how to report the search. But this is only technicalities, let's say. Then there is the most important part, how to summarize the information that are retrieved in the search. So the applicant is requested always not only to give the panel the list of publication, but also to summarize them. So it is required to have a summary table which summarize the outcome of the selection process. This should include a minimum set of information. So it means obviously which is the additive, the test item, the product which is reported in all the different studies considered relevant, the concentrations, the animal species, the duration, so all the minimum set of information necessary, which endpoints are assessed, and also the summary of the different effects which have been observed. So these for each of the study which was considered relevant after applying all the excluding criteria. And then last but not least, the applicant is requested to provide a critical assessment of the evidence collected. So not only a list, not only a table, but also to try to present the results with a critical assessment of the evidence and possibly a description of the results and a conclusion drawn by the applicant or who is performing the literature search. Having said so, we can say also that so far we had a bit of experience with some literature searches sent to support the renewal process, and we have identified that there are some recurrent issues which would not allow to consider the literature search, at least as a first step, as a complete set of information. And these may be divided mainly in 4 big areas. The first one is the one related to the time span. So as we said, probably not covering all the time needed or not finished 1 year before the submission, but well in advance, and all possible issues related to the time of the search done. The second main areas is related to the characterization as the additive. As we said, for example, the search is done only with generic names, but the trade name of the brand name is not included, or only one of the possible name was looked for and not possible synonyms or other names, or for example, as we said, for mixer, only the most relevant parts included in the search, but not the whole mixture or other components of the mixture. Then the third group is related to reporting. As we said, it's highly important to report correctly what was done. And it happens that the search terms are not reported, or the strings, or the combination, which excluding inclusion criteria were applied for, and so on. So this will not allow, or did not allow, to evaluate properly the results. And the last one, it's related to the different sections of the safety which are not included in the search. For example, the search is limited or focused on safety for the target species, but not on user safety or environmental safety or safety for the consumer and so on. So this will trigger obviously the need for additional information. As I said at the beginning, there are two main ways to submit or to provide this additional evidence. One is the extensive literature search that we have seen now. The other one is the possibility to submit or to provide the new studies. This possibility is always given to the applicant for any aspect of the safety of the additive, possibly also of the efficacy if necessary. So it's really up to the applicant to decide whether new studies might answer better the question on the safety of the additive. However, it's important to keep in mind that new studies, also in this case, should absolutely respect three main characteristics. And these are that the studies should be completed not earlier than 3 years before the submission, because since the scope is that the additive remain safe as it is on the market now, older studies could not give a proper picture. They should reflect the current farming or production condition in Europe for the same reason. And then, last but not least, they should be performed and reported, obviously, according to the relevant guidance document on the different section on the safety or efficacy of feed additives, the guidance mentioned by my colleague Jaume before on the different aspects. Now, since we have seen how this additional evidence, this general additional evidence could be retrieved and submitted, we can look a little bit more in deep how this applies to the different sections of the safety. But in particular, on the fact that, as we said, these are the minimum requirements that are requested. So to retrieve information, to look in the literature if any adverse effect has been notified or identified, and to provide this to the panel. In addition, for the different sections of safety, there are some specific issues which should be taken into account when setting up the dossier for the renewal. For all the different sections, you will see its recurrent aspects to be considered is that there are exclusion criteria. It means that there are sections in the relevant guidance, for example, in this case, in the guidance for the safety for the target animals, which lists for which additive no additional information are required, because the additive is considered is given already as safe, and therefore no additional evidence are required. So this is probably the first step to be considered and the first aspect to be checked. Then, if the additive is not in this list, then more specific details are to be considered. In particular, regarding the safety for the target species, you know that when an application was done for all animal species, it was sufficient to demonstrate the safety in 3 major species. And according to the older guidance, these 3 major species could be terrestrial or that could be fish. If the application was, the authorization was granted based on studies in 3 terrestrial major species, then now it is requested that additional evidence is provided to support the safety also for fish, in particular salmonids. This evidence should be, as we said, in the form of literature search if any information is available. Otherwise, new studies would be required. The second issue to be considered instead is when an authorization was granted with the maximum content in feed that was derived applying a calculation to calculate, as I said, the maximum safe concentration in feed starting from a NOAEL or a BMDL identified in toxicological studies. So in this case, what is requested is to verify if new or different NOAELs or BMDLs are available or became available since the last authorization. And if this is the case, then a new calculation using the tool available on EFSA website, this calculation, as I said, should be redone to check whether the maximum concentration will remain the same or will change if a lower NOAEL or BMDL is identified from the available information. The last 2 aspects to be considered for the safety for the target animals comes from a possible modification of the composition or the manufacturing process, as Jaume said in the specific section. Some of the modification could have an effect on the safety for the target animals. For example, change of the composition or the manufacturing, which would modify the bioavailability of the active substance, could have an implication on the safety for the target animals, and this should be addressed or at least specifically considered. And the same is to be done if any information, any evidence of a specific adverse effect on the target species became evident in the literature or became available in the last 10 years. More or less the same initial approach is to be applied for the safety for the consumer. It means that also in this case, additional evidence we have seen how to retrieve them are the basis for the demonstration of the safety for the consumer. In addition, few additional information should be checked. It means should be verified. And these are any new data or information on the ADME of the additives, so absorption, distribution, metabolism, excretion, and even deposition. If any new toxicological properties became available, and obviously also exposure of the consumer to residues could be taken into account if any new evidence became evident in the last years. So also in this case, there are exclusion criteria. So all those additives which are listed in section 3.1 of the respective guidance are excluded from the need for any additional information. For all the other, instead, some specific issues should be taken into account. And we can start from the residue. So it means that in case in the authorization it is requested to monitor the residue, the results of the monitoring should be always submitted in the dossier. If there are evidence that the metabolism in the target species could be different from the metabolism, so it means that the metabolites or the residue could be different from those which were evaluated at the time of the first assessment, this should be taken into consideration. The same as we said before, in case of modification of composition of manufacturing, which might lead, for example, to effects on bioavailability, for example, increased bioavailability of the active substance of the additive, or if any additive for which an health-based guidance value was established, is important to check that these health-based guidance values remain safe, or to check for residue if in the meantime a new health-based guidance value has been established by any evaluating organism. Regarding toxicological data, like for residue, there are few situations in which it could be needed to give more information or to put a specific attention. And these two cases mainly are when new information became available in these last 10 years, or when as I said, the assessment of the additive or the active substance from any scientific body led to a different health-based guidance value, or establishing an health-based guidance value compared to the ones already existing at the time of the first evaluation. So in this case, specific information might be needed. And then there are a couple of specific issues regarding the genotoxicity potential of the additive that should be taken into account when preparing the dossier. The first one relates to those applications in which the absence of genotoxicity potential of the additive was established using a UDS test. Since this test is no longer recommended as an in vivo follow-up, it means that if the study did positive results, okay, these are considered adequate and could be retained. If instead the study did or gave negative results, then the reliability of the results should be evaluated in a weight of evidence approach, considering all the other information. And it could be the case that a more sensitive test could be necessary in this case. The other issue to be taken into account is instead when the absence of genotoxicity was demonstrated with in vivo tests which gave negative results, for which it's important to check that the exposure of the target tissue was present. If not, also in this case, additional evidence or additional data could be needed to demonstrate the absence of genotoxicity of the test item or of the additive. And then, last but not least, remaining in the genotoxicity testing strategy, when talking about chemical mixtures, complex chemical mixtures, for those mixtures which could be fully characterized and identified, then a component-based approach should be considered. So it means that all the available information should be used to support the safety, so to verify the possible genotoxicity of the different components of the mixture. The read across could be applied, cursors could be applied, but it's important to be able to demonstrate the absence of genotoxicity of all the different components. In case instead of a chemical mixture in which some of the mixture cannot be identified, chemically identified, then a slightly different approach should be taken. It means that for those parts of the mixture which could be identified, then the same approach remains, so they should be assessed individually, but then also a testing of the whole unidentified fraction, or even if it's not possible, of the whole mixture should be considered. Then, again, remaining in the consumer safety part, also few tips for exposure. As we said, if the additive is characterized by an health-based guidance value, then consumer exposure should be reassessed, should be re-estimated using the most recent data and following the instruction given in the guidance on the consumer safety. Sorry. And in particular, also information on residues in food of animal origin, also from other uses as feed additive, should be submitted, and if possible and if available, also exposure of the consumer to residues by sources other than food of animal origin. Obviously, this only if available and if retrievable. Then, also regarding the safety for the user, there are a couple of issues to be taken into account. So there are some aspects which should be always submitted or considered when relevant. So it is requested to put in the context of a real use of the additive. So it means that if there is any record of adverse effect in the manufacturing plant or to person exposed to the additive, should be provided in the form of an extract of the complaint management system. Or if the additive was already evaluated under other regulatory frameworks, like the REACH one or CLP or any other regulation, also the results of this assessment should be provided. And the last two issues to be considered are that if the database, the original data set was not complete, and therefore the opinion on the panel in the past could not conclude on all the aspects on the safety for the user, this data gap should be filled. So a full set of data would be needed. And as we said at the beginning, if the manufacturing has changed, then any possible implication of the change to the safety for the user should be taken into account, in particular considering possible changes in exposure and potential new hazards introduced, modifying, for example, carriers or any other component of the additive. Then, last but not least, also the safety for the environment, as we said, should be taken into account. Also, in this case, with the literature search, if this is the strategy chosen, this should be included. But some also issues should be considered in this case. There are some specific criteria which would exclude the additive from the need to provide any additional evidence, and these are all listed in the guidance. Otherwise, if the additive cannot be excluded from the assessment following these exclusion criteria, then we invite the applicant to check with the guidance on the safety for the environment, the new one adopted by the panel, and to check if under the current requirements, so those included in the guidance, the additive would need new data or new information to give a full picture and to demonstrate that it remains safe also for the environment. Also, in this case, from the experience that we have gained so far, we have identified that there are some recurrent issues when assessing the different aspects of the safety. I divided them for the different sections, but just highlighting one for each of them, just to give you a picture. But there are many others which trigger the presentation mainly. So the most important ones that we have seen so far, for example, for the safety for fish, no information are submitted at all. Because, for example, nothing was found in the literature, but keep in mind that, and this is something that could be applied to the whole process, not always the absence of evidence is evidence of absence. So sometimes it's not sufficient just to say there is nothing. So everything should be put in a context or clearly justified. Regarding the safety for the consumer, one of the main and most recurrent issues is that exposure estimate is redone, recalculated, but not considering all the available residue data. So maybe only the old ones and not the new ones, or the opposite, or not all the different food commodities are considered. Regarding the user safety, the old database, the original one, the old data set, sorry, is submitted exactly the same of the original one, so including the gaps which were present in the original one. So this will not allow a full assessment. Or, for example, for the environment, again, the previous demonstration of efficacy is resubmitted, but we're not checking with the current requirement, and so missing some information. So we strongly invite you really to check the different guidance at any moment to see if you are fulfilling all the different requirements for the different sections. Now I have still one slide for the last part of the guidance, which consider efficacy. You have seen since the very beginning that the main focus of the renewal is on safety, but a few words are important also for efficacy, because normally it's not required for the renewal unless the applicant is requesting for some amendments of the terms of authorization, as Jaume explained at the beginning. In this case, efficacy study might be needed, and this should be done in compliance with the current requirements, it means the current guidance. The only case in which instead efficacy studies are really needed are for applications for renewal of cocidiostats or histomonostats. In this case, evidence of the maintained susceptibility should be provided, and in the guidance the details are given, for example, on the age of the strains, the identification of the strains, the origin, so at least three, it means 3 different studies, which kind of studies should be done, so sensitivity studies, and the fact that the studies should be completed at least 2 years, so not more than 2 years before the submission, for the same reasons that we have explained before. So just to wrap up a little bit what we have said so far and what was the scope of the preparation of the whole presentation. First of all, please always check in deep the guidance on renewal. Check all the different aspects, the exclusion criteria, but also the inclusion ones, and all the different issues and aspects which could be important. Try to follow the structure of the section when preparing the dossier for renewal. So following the guidance and preparing the dossier, try to follow it. It will ease your work and will ease the work of the panel when assessing the information. Be sure to include always all the relevant information. So all the study reports, the raw data, certificates, previous scientific opinion of the panel or EFSA or any other body, please include them all. Be sure that all the data that are cited in the dossier are always included. As Jaume explained at the beginning, be sure to clearly identify, even using color, highlighting a different font type, whatever could be a good way to identify what is new in the dossier and what was previously submitted. So in this way, it would be easier really to distinguish the two sets of data and to perform a proper assessment. And then, last but not least, try to follow also all the other guidance which I and Jaume mentioned this morning, or which are referenced in the guidance document. So with these last tips and good practices, we have finished a presentation on the guidance. Now we open the session on question and answer. I give the floor now to Frank, and I thank you very much.
Thank you, Matteo and Jaume, for the presentation, and also to keep perfect track of time. We are really on schedule for that. So it means we have a lot of time also for the question session. We will start this with first the reply to the written questions that we have received from some stakeholders before the meeting. Then we will go through the questions that we receive in the chat box, and then if we still have time, then we can also ask if there are some additional questions or have a brief discussion. So let's start with the first question that we received before the meeting from Mary Sutinen from BioSafe, and the question was: is the sensitivity limit for testing for the presence of DNA in the product testing changing from 10 nanogram DNA per gram or milliliter products? To reply on this question, I would like to ask Baltasar Mayo. He is a member of the FEEDAP panel, and he is also the chair of the microorganism working group. Baltasar, floor is yours.
Okay, thank you, Frank. And thank you to Mary Sutinen also for the question. I think it's very easy to answer. In short, the answer is not. The sensitivity limit of detection of DNA is not expected to change soon. The threshold is a convention. As you may know, this 10 nanograms per gram or milliliter of the product, and was established based on the experience accumulated through the assessment of many dossiers, and after long discussions with the European Research Laboratory and many experts on PCR. So the FEEDAP panel and the FEED unit apply a careful vigilance of the suitability of all the requirements established in the guidances, and as such, these limits will be reviewed for sure during subsequent updates of the microbial guidance. But at the moment, the applicability of this specific threshold does not indicate a need for modification. So we don't expect any change for the moment. Thank you.
Thank you, Baltasar. Then we proceed with the second question from Anouk Lankriet. Would EFSA consider to only publish the reference list of the cited literature instead of the full scientific publication to avoid high copyright costs for the applicants? So I will reply myself to this question. The applicant must ensure that the terms and the conditions asserted by any rights holder of studies, information, or data submitted to EFSA are fully satisfied. The applicant may consult the copyright licensing orders. It is, for instance, at national level for guidance on purchasing the appropriate licenses to provide studies, information, or data to EFSA, taking into account the proactive disclosure requirements as detailed above. For publications already available to the public upon payment of fees, such as studies published in the scientific journals, for which the applicant does not have or cannot obtain intellectual property rights for the purpose of the proactive public disclosure requirements, the applicant must provide a copy of the relevant publication along with the relevant bibliographic references and citations for scientific assessment purposes only in the confidential version of the application. And secondly, these relevant bibliographic references or citations where these publications are available to the public, these should be in the non-confidential version of the application for public dissemination on the Open EFSA portal. Then we can move to the next slides and the third question from Mikkel Kroni. What are the implications of the transparency regulation on the renewal of the authorization of feed additive? I will also reply on this question. It is recommended to consult the 3 practical arrangements that are published on the EFSA website. And these will specify also how the transparency regulation will be applied by EFSA. For the renewal processes, the main new steps are the notification of the studies, the public consultation on the attended studies, and the provision of the renewal pre-submission advice by EFSA. The new rules on the confidentiality and the transparency also apply. So all the information is available on the website, and as we said in the beginning, transparency regulation is not the main objective or not within the scope of this meeting. Then we can go to the fourth question submitted by Sarah Roman. On average, how long usually could take the whole authorization from the submission of the dossier to the publication on the feed additive register? To reply to this question, I will ask Gloria Lopez Galvez. She is a senior scientific officer in the feed unit. Gloria, floor is yours.
Okay, thank you, Frank, and good morning to all. I am a scientific officer also in the feed unit of EFSA. Okay, thank you, Sarah, for your question. Indeed, the whole authorization process has, let's say, 2 main parts. And the first part is in the hands of EFSA, and the second part is in the hands of the European Commission and member states. If we focus on the EFSA part, it covers from the receipt of the dossier until the opinion is published. And in this part, we have at its own a sub-part that is on the hands of application desk, and application desk performs a completeness check of the dossier. It takes a maximum 30 working days. Then from that day in which the dossier is complete, the assessment, the maximum time allowed by legislation is 6 months. And well, during these 6 months, EFSA could also ask for supplementary information. So in general, yeah, the more complete and the less supplementary information have to be requested, the faster is the risk assessment, and it can be completed in less time. Then the second part of the authorization, as I said at the beginning, is in the hands of the European Commission and member states. And this goes from the date in which the EFSA opinion is published until the regulation is voted. In the legislation in the 1831, it is indicated that that the European Commission has 3 months to draft a regulation since the receipt of the opinion from EFSA. So, well, now also I would like to say that, as you all know, there is a new era in EFSA with the application of the transparency regulation. And so we will, of course, try to stick to our obligations and to the time that EFSA has allowed for the assessment.
Thank you, Gloria. Maybe you can stay on because also the next question will be replied by you. Question submitted by Sandra Mattoni. Question is, will there be additional technical meetings?
Okay. Thank you, Sandra, for your question. Indeed, these technical meetings are held to boost the engagement with the stakeholders, and in our case, also in the FEED case, to make the applicants better aware of the EFSA guidance and the technical procedures. This year, it will be this one that we are currently holding, and then there will be also another meeting with a stakeholder, with applicants, open to stakeholders. That will be the open plenary meeting that will be held in November. The dates are 9 to 11, and yeah, we will state which will be the date for the open plenary. So at the moment, we from FEED unit are not foreseeing any other technical meeting for this year, but my colleagues of application desk may hold a technical meeting based on needs of applicants. And so for that reason, the colleagues from the app desk are now collecting needs. So then if you have a specific request, you can send your message and your email to us via the different sources that we have, or asking a question with the online form, or contacting us. And also another possibility is that you get the newsletter and the email alerts if you subscribe via this Connect tab in the EFSA website.
Thank you, Gloria. Maybe just wanting to add regarding the transparency regulation, that there are a lot of trainings, webinars, videos available on the EFSA website that also might be really of interest to the stakeholders and can be consulted. There is also a calendar available of further upcoming trainings. For instance, there is one coming up, I think tomorrow around lunchtime, on confidentiality assessment. Thank you, Gloria and Baltasar, for supporting and replying to the questions that we received before the meeting. So now I think that we can go to the questions that we received during the presentation in the chat. Let me just scroll to find here the first question. First question was from Ruud Bremmers. Can the quantitative data on the use of the additive be kept confidential? Well, here it's the responsibility of the applicant, of course, to submit the claims for the confidentiality to EFSA. This is something that changed with transparency regulation. It is now EFSA that has to assess the claims, and the claims should be funded on the legislation. So the 1831 from the feed additives, but also the general food law, and there you can consult Article 39C on commercial sensitive information, if it can be protected or not. I would recommend, if you have a specific case, maybe to submit your detailed question to the functionality of Ask EFSA, and then colleagues from the legal unit can look into that and provide specific advice on the question you might have. Then we can go to the second question from Manfred Lutzow. question on the geographical distribution, whether it is only in the European Union or globally. Maybe Jaume, you can provide some clarification on this question.
Yeah, sure, Frank. Yeah, in principle, the preferred data is the one produced in the European Union, so the volume of the additive that it's sold and used in the European Union. But if there is data also on the global distribution, this is also fine. Thank you.
Okay, then we can go through the third question. If one of the questions would not be completely clear in the reply that we provide, at the end we will open again the floor, so then you can come in to have additional questions if required. Question 3 from Caroline van der Schoor. Is the literature search an optional or mandatory part of the renewal dossier? Maybe Matteo, you could try to reply this question.
Yes, thank you, Frank. Yeah, thank you for the question, Caroline. No, as is reported also in the guidance, the literature search is a tool which can be used to retrieve information. Is not mandatory, is one of the possibility. The other one is to generate studies or to provide information by other means. This is suggested to be the most inclusive one, so that's why it was so detailed, but it's always an option.
Thank you, Matteo. Then we move to the next question. It's again from Manfred Lutzow. We said in the beginning just one question per stakeholder, but as we don't have so many questions, I think we can just proceed and also look at the other questions from Manfred. So the question goes also on the literature review, as most feed additives are multi-purpose substances like vitamins, which make the work quite intensive without providing significant papers. Any views how to provide more relevant literature reviews? To reply on this question, Jordi Terescales, I would ask to explain this question, and he is also scientific officer in the feed unit.
Yes, thank you for this question. We recognize that extensive literature searches in some cases can become complex and result in an outcome of relative relevance. There should be a balance between sensitivity, trying to detect as many papers as possible, and specificity, trying that the paper retrieved are relevant to the review question. The search strategy should be well elaborated in an iterative process. Having a bunch of relevant papers ad hoc that you know should be retrieved by your search and checking if they are captured by your search strategy, this might help. Of course, playing with search terms as the Boolean operators, the and or, and also the not. There are search functionalities in the different databases as truncation, wildcards, proximity operators, search limits and filters that help to fine-tune the search. In complex situations, the support of an experienced librarian could be very valuable. And just to finalize, there is some guidance on extensive literature searching indicated in the guidance on the renewal. That's all.
Thank you, Jordi. The next question is also from Manfred. For an essential oil, I would have to perform 100 literature searches for each constituent, separate search. To reply to this question, I would like to ask Paola Manini, who is Senior Scientific Officer in the feed unit, to provide clarification.
Yes,
Paola.
thank you, Frank, and thank you, Manfred, for this question. Of course, we don't expect to have 100 literature searches for an essential oil because we are gaining experience with the assessment of this oil, and we are seeing that mainly there are few components of the oil that are driving the risk assessment. So we want to have information on those compounds, the major component driving the risk assessment, and also substances of concern, because the full characterization of the additive in this case is aimed at identifying substances that maybe could be genotoxic or maybe could have some special toxicity. In addition, I would say that many of the components of the essential oil are also evaluated as chemically defined flavoring, and the renewal of these substances will anticipate the renewal of botanicals. So I think that also in this case we could benefit of the renewal of chemical flavorings for the renewal of botanicals. And also I think that it is true that these essential oil are made of 100 components, but some of these components are also common across the different essential oils. So this information can be also used horizontally for the different preparation, especially if there is a consortium of applicants behind.
Thank you, Paula. Then we have 3 questions on the same topic. It relates to the 3-year limits for the studies before the dossier will be submitted. Question from Manfred, from Ludovic Arnaud, and Caroline van der Schoor. Jaume, I would like to ask you to provide clarification.
Yeah, sure. I think that the 3 questions refer to this limit of the 3 years, so that studies, when the applicant decides to submit additional studies for the demonstration of safety, this should be done during the 3 years before the submission. I think that the first thing to clarify is that this is not a requirement, so we are not asking applicants in any case to submit newer studies. This is one of the options that Matteo explained, in which you can provide evidence of that the additive is still safe for the target animals. This applies only to studies done for the target animal safety, so not for efficacy and not to toxicological studies. And I hope that this is clear. At least we try to clarify that during the public consultation and also in the guidance. It's clearly expressed that this applies only to the safety for target animals. The reason basically to why we limit it to the last 3 years is to ensure that the conditions of the study reflect the most recent production or current farming practices in the European Union in terms of breeds, diets, husbandry, and so on. So that we ensure that the most sensitive animals are used in these, let's say, new tolerance studies. As I mentioned, for efficacy, there is not this requirement, so that requirement does not apply, but the efficacy studies are only required for coccidiostats. This is the only case in which there is a requirement for studies. And in that case, if you add the 2 years plus the 3 months before for the isolate, we are more or less again also at the 3 years time frame. So I hope that this clarifies the 3 questions.
Okay, thank you, Jaume. Then the next question is also from Manfred. Is the additional genotoxicity testing also needed for food flavorings which are not subjected to the risk assessment when used in foods? Maybe, Manfred, I would like to ask you to clarify a bit more the question. So if the technical colleagues could give the micro to Manfred, and then Paula will try to reply. Manfred, are you able to explain the question a bit better? Yes, come in, Manfred. I hope you can be unmuted.
Okay, can you hear me?
Yes, we can hear you.
Yeah,
yes.
Good. Yeah, good morning. Thank you very much for allowing me to explain that a little bit. The point is that in the area of food flavorings, we do have the principle that flavoring extracts can be used in food if they have been if they are derived from sources which were in use when the legislation was adopted. That's a principle. So you do not need to subject natural flavoring complexes to risk assessments, including genotoxicity testing and so on. In addition, we have a number of substances of concern which are limited by maximum levels in food, some of which are genotoxic compounds. But most of the flavoring complexes used in animal feed are used in food flavorings, and it is always a difficult discussion and consideration to start full-scale genotoxicity testing for substances which are used in considerable amounts, partially in food, because you of course have a kind of potential effect on the safety considerations for human food. So it's around this my question. I have a waiver on the food side. For consumer safety, this waiver does not apply when the same substance is used in animal feed. Is that correct?
Okay, Paola, if you could reply.
Okay, I can try to reply because we are aware that there are differences across the different sectors. But, for example, for this extract of natural origin, there were limits that were based on practical consideration more than on scientific assessment of the genotoxicity and toxicity of the substances. And also these are based on the fact that there is a traditional use of this preparation for humans, which is not the case for animals. So in principle, we have to follow our rule, and we are trying to perform a risk assessment based on the rule that we are continuously discussing also with the colleague in the food area, because for these substances we are also taking into account the implication that the conclusion of the risk assessment for feed additive could have on the food area. But in principle, yes, we cannot, we have to perform a risk assessment, including the genotoxicity also for this preparation. We are trying to set the methodology that is under discussion in the panel, but also inside EFSA with also the other panel and the relevant working group, horizontal working group that were set at the EFSA level. But remember that this, the fact that the intake of substances of concern, okay, we humans are having this intake, but it is not, does not mean that it is safe. And also the idea is to avoid that there is an additional burden, an additional intake from the food of animal origin following the use of this preparation as feed additive.
Question is also from Manfred on the user safety. Most feed additives are covered by REACH or CLP. Could you comment on the relation of work done by EFSA and ECHA? I think Fabiola will provide a reply. Fabiola, also a scientific officer in the feed unit.
Yes, thank you for your question. Yes, indeed, it is possible that a feed additive is also covered by other regulatory framework, and in case a feed additive or a component of the feed additive has been assessed by ECHA, the applicant can also provide the outcome of this assessment, but in any case it should be sure to have the rights to use the data, as they can come from a different applicant or a different company.
Okay, thank you, Fabiola. Then next question is from Ludovic Arnaud. Fish safety and Silas products. How do you see this, as the most Silas additives are authorized for all species, but Silas additive mainly used in terrestrial species? Matteo, could you provide a reply?
Yes, Frank, thank you, and thank you, Ludovic, for the question. It's true that most of the silage additive had studies, if any, done with terrestrial animals, but it's also true that the vast majority of silage additive made of microorganisms are based on QPS microorganisms, for which demonstration of safety for the target species is excluded anyway. And for those which are not QPS, that do not qualify for QPS approach, there are several exclusions also in the guidance for the safety for the target species, some characteristics which would allow them not to do additional studies. They are listed there. You probably know the fact that, for example, the additive is not present in the silage in extensive amount at the end of fermentation, or they do not they cannot be distinguished from those naturally present in the silage, and so on. So there are really many possibilities. If also considering all these possibility to exclude the need of further information, then there will be the issue of the fact that we will remain with 3 major terrestrial species, and you will need to confirm the authorization for all animal species. Then information of fish would be anyway needed, but this will be really a minor amount of additives which are authorized as silage. So far, at least with those that are authorized, this is since this is a renewal. So for those which are authorized, this is the situation. So I hope to have clarified the question to Ludovic and all the other. Thank you.
Thank you, Matteo. If not, there will be the possibility afterwards to raise a follow-up question. Let's move to the next question from Ruud Bremmers. What if the organization originally submitting the dossiers does not exist anymore? Well, it's the applicant for the renewal who may need to make sure that the data needed is available to EFSA. I don't think we can add a lot more to this question. Next question is from Manfred. Main question, your underlying assumption is that the application is the same, which is often not the case. Manfred, again, can you maybe clarify a bit more the question? It's not that clear to us.
Yeah, sorry. It is basically the same what Ruud Bremmers is asking, and it's I wrote application. It should be the applicant. The slides from Matteo about the characterizing what is old and what is new in an application reflects the understanding that the same applicant applies. Now, this is in some cases true, but in other cases, specifically for generic additives, it's not necessarily the case. So I wonder where this assumption is coming from. I would have expected that a renewal does not, for a generic additive, a non-holder specific additive, does not work on the assumption that you have a continuous applicant doing the first and the second one. I'm a bit surprised about that assumption.
Yeah, I'm not sure, Jaume, if you could provide some view here.
Yeah. I think Manfred, you are absolutely right. It does. I think what we request in this fact to identify the data that was already submitted is for those cases in which the applicant is the same, and that will be basically the case for zootechnical coccidiostats, probably for several also nutritional additives produced by fermentation, in which even if they are not generic, they are not brand specific. Let's say they are at the end linked to the owner of the strain. In all other cases, if this is not possible, of course, we will not ask applicants to identify what was already submitted by someone else. But in those cases in which the applicant are submitting the same data as it was submitted originally 10 years ago, we ask you to identify that because it helps us very much to process the application. In other cases in which the original applicant does not apply for the renewal, then it is up to the new applicant to provide all the data. And in that case, it can be all new data, or they can try to find data from the past applicant if that's still possible. I think that's, and we have seen that already in some occasions.
Yeah, thank you. That answers my question.
Thank you, Jaume. And then we come to the last question that we have in the chat from Didier Jans. First slide indicated that renewal is not a reauthorization. For many dossiers, the original one might be quite far from the current requirements. Have you made some pilot to evaluate such gaps and estimate the resulting burden in a kind of impact assessment logic? Also for this one, I'm asking Jaume to provide some clarification.
Yeah, sure. I'm not sure if I understood your question right, Didier. So in case that I did not, just you can clarify later on. But I mean, we have not done really an impact assessment of the changes introduced in the guidance. In any case, we are not asking that the new dossier complies with all the requirements from the new guidances. I think that what we said at the beginning, the additive has already been assessed for its safety 10 years ago, or let's say 10 years before the renewal. So we do not, what we ask you is just to provide evidence that it remains safe. Only in very few cases, and I think that applies mostly to the microorganisms, to the genotox, and some data gaps in the user safety, we ask you to address the discrepancies, and the environment. That was the maybe exception. We did some check when we had this in mind. For example, for the issue of the genotox, we went back to the original dossiers and we saw that there are not many in which there is this UDS or the requirements were so different to the ones that we are applying now. So I understand your concern, but I don't think that this is really such an issue. In any case, we have a clear task, which is to ensure that the additive that is in the market remains safe. So we need to make sure that the assessment is performed up to a good scientific standard. I don't know if that addresses your concern, Didier.
Yes. Do you hear me?
Yes.
Thanks, Joe. Yes, of course, I fully understand. Well, safety is at stake. It needs to be covered. I'm a bit concerned on a number of dossiers. You know, for instance, when you look at the extensive literature search, that's not the kind of thing that has been done in many of the dossiers. If you start with that, you enter a new field compared to the initial dossier. And even though if you do not apply completely the new guidance, this might, at the end, well, at least that's my guess, represent a very significant step and burden for a number of applicants.
Yeah, I understand that, but again, I think that's not on our side. Let's say our task is very clear, is that we need to make sure that the additives are safe at the end of the day. Yeah.
Fully appreciate. Just raise the concern.
Sure.
Yeah.
I understand. Thank you, yeah.
That's clear, and I think also the reply from Jaume was clear. Okay, so with this, we ended the list of questions that we received before the meeting and also during the presentation in the chat. Are there any additional questions? You could write them in the chat or ask for the floor. We still have a lot of time. Don't see anybody raising the hand or asking for the floor. Yes, the slides and the registration will be available on the EFSA website. Yeah, so that also participants that could not subscribe for the session today, or stakeholders, have to say, can also look at the information. I think that is one of the advantages on having a live virtual event, that we can also ensure that all the information is shared later on with all the interested stakeholders. I would have a question on the whole genome sequencing. Yes, Manfred.
Yeah, thank you very much. Sorry for taking again the floor, but I appreciate this framework here and the way you are running the webinar. So my question on whole genome sequence is that for the renewals, many of the organisms were not, let's say, characterized by whole genome sequence 10 years ago. And of course, the methodology has improved and changed. My question basically is, how many whole genome sequences do you want to have in order to characterize the organism? We had cases where we had to submit several ones for the same organism, and this is related to the point that a microorganism, even if I have a stable population, may fluctuate in the sequence of the bases in the DNA. So, in addition to is there a bit more close definition? What do you want to see as whole genome sequence? Just 1 year before the submission, or several ones? The other question is, what actually is the whole genome sequence that characterizes a strain? Does that allow for smaller deviations? Let's say a base pair change. Is that already creating a new strain? When does a new strain starts, in your view, when you characterize a strain using whole genome sequence, which is a different thing as we characterized strains 10, 15, 20 years ago? Thank you.
Yes, Manfred. I will ask Balthazar Mayo, chair of the working group on the microorganisms, to provide a bit of clarification to your question. Balthazar.
Okay, thank you, Frank, and thank you, Manfred. I think that the whole genome sequence is a tool, and is a very sophisticated tool to help into the identification first, and then into the characterization of the production strain or the strain under assessment. We know that the strains are changing over time, but what we need is just to use this whole genome sequence for assessing the safety. So once you have the genome sequence, although you may have some changes in the future, that will be definitely if there are no problems, if there are no issues of health concern. So we are aware of small changes through the genomes and so on during the time, but I think the whole genome sequence is used as a tool to identify the strain and to characterize the strain.
Thank you, Balthazar. Maybe I also ask Montserrat and Greta Freixa to provide additional information on the second part of the question, and also maybe make a reference to the EFSA statement that was published recently on the whole genome sequence. Montserrat.
Hello, good morning. Thank you, Frank. Thanks also, Manfred, for the question. Indeed, regarding the second aspect as to that regards where the strain starts, no? I would say that, as you know, we normally ask you for the history and for the origin. So the origin and then the history of modifications that you do with your strains, because that's important, as Balthazar said, because that may trigger a modification on the safety of the strain that you are using. So whenever an applicant has done a step of modification, which can be then after deemed as a genetic modification or not, no? A step of mutagenesis, for instance, which would not be as a genetic modification, but that would need to be declared, because that may result in a different strain, no? And yeah, regarding the thing that in the past we didn't used to use the whole genome sequencing data, that's clear, but the panel also considers that it's a powerful tool where we have all the information for the strains, and it's the way forward. And in fact, the panel has worked together with other experts from EFSA to provide you with a better guidance in the statement on the whole genome sequencing, which has been published in the web, and which I hope that you reached, and in short will be published in the journal.
Thank you, Montserrat. I hope, Manfred, the reply is clear to you.
Yes, thank you.
Then the next question is from Guillermo Jimenez. Why the extensive literature search to include in the renewal dossier is limited for the period comprising since the last assessment until not more than 1 year before the date of submission of the application for renewal? Why not allowing the inclusion in the dossier of literature published until the day of submission of the application? Matteo, could you provide a bit of clarification on this question?
Yeah, sure. Thank you, Frank, and thank you, Guillermo, for the question. Maybe the way in which I presented it, it was not clear, and therefore I created some misunderstanding. The time span is not absolutely limited to 1 year. It means that it should not be done before 1 year before the date of submission. So it should mean not older than 1 year. You can do it until the last day, absolutely. That was a compromise to limit to 1 year, because then if any issue would arise from the literature search, then you will need to have the time to generate the additional evidence that would be needed. And since there is a legal limit for the application and the submission for reevaluation, for renewal, which is 9 years after the authorization, so 1 year before the expiry of the authorization, this is to give the applicant the possibility to retrieve and collect any additional information if needed before the expiring. But as I said, you obviously have the freedom to do the literature search even the same day in which then you do the submission, having in mind the fact that if anything is then highlighted in this literature search, you will need then to retrieve additional information. The scope is that we don't want to have literature searches which were done more than 1 year before the date of submission. I hope now it's more clear the time span needed, and the answer clarified your doubts.
Thank you, Matteo. Don't hear question from Guillermo if he needs further information. Then I invite also still the other stakeholders to submit additional questions if you have something that needs clarification to us. Wait a couple of moments to see if something else is still popping up. Okay, we have somebody asking the floor. It's Roberto Villa. He is also panel member. Roberto, floor is yours.
Could you hear me?
Yes.
Okay, so I'm Roberto Villa from the University of Milan. And no, I would like just to say something about the literature research, because I think that is quite important. Maybe they all know quite well how to do it, but because also in the university we use quite often to do the literature search. And I have to say that it's not maybe just because I'm stupid, but it's not so easy. So because we have different database, and the rules of the database are quite different. For example, CAB abstract or Medline. And so my suggestion is that when I try to do it, it's not easy to retrieve the right literature for the task that you have. So I suggest that it's quite important to use professional people working from the library. They know all the language of this database. and sit on the side of these people trying to explain what you want and to focus on that. And furthermore, I think it's really important then to be sure that you are able then to obtain all the complete paper, because quite often we receive just the abstract and then we have to go back and ask for the other part. And after that, if you focus quite well on the different subjects that you have to cover for your literature search, then it's very important also how to present this data and resume what is really important. So I think that it is important, because quite often when you try to do the literature search for important substances that are quite studied around the world, you can find, you know, thousands of papers, and that's make our job then very difficult if it's not well focused and synthesized and summarized at the end. So that's I think is an important suggestion. Maybe you know very well this this problem, but I think it's quite important to underline this, I think. Okay, I finished.
Thank you, Roberto, for this good suggestion. I think there is also EFSA guidance available that can be retrieved from the website that is available on literature reviews. In the meantime, I don't see any other question that appeared. We also have colleagues connected from the European Commission. If somebody from Commission colleagues would like to come in, make a statement, that is fine. Otherwise, I think we will proceed with closure of the meetings. Don't see further question coming up. So then I think we are coming near the end of the meeting. I hope this session was really interesting to you, to have additional clarification on top of the guidance that was available already to you during the public consultation. Also, some of the stakeholders attended the open plenary in November when there was the last discussion and the adoption. Today, yeah, we also tried with this session to clarify additional questions that you might have on this topic. So we believe that is important for you as applicants to really read this guidance carefully and apply it as good as possible. It looks obvious, but it's not always done very carefully. And I think it's in the interest of all of us to have the dossier as good as possible, because then we can proceed fast, which is also in the benefit of applicants to have the conclusion of this process as soon as possible. As stated several times during this meeting, a renewal is not really a reassessment, but we need to just ensure that it's a second check on the safety, not regarding the efficacy. So the requirements, we believe they are not that many and also not complex, because we agree that the focus is the assessment whether the product is still safe. And with this guidance, we concluded a series, as already explained by the chair of the panel, Vasileios, in the beginning of the meeting, and also by Jaume in the start of his presentation. We conclude a cycle of updating of several guidance documents. Over time, we will gain experience with this guidance and also the other ones, and for sure then there will be a new cycle, starting whenever there is a clear need to update one or more of the guidance documents. I think we are also living in exciting periods with initiatives, for instance, from the European Commission on the Green Deal, where we also expect updates on the feed additive regulations. So it is likely that in not so far away future, we might need to reopen some of the guidances, and then we will again consult all of you, because this work is not just done by EFSA alone. We started this whole process with also having a meeting with you as stakeholders to collect some input, some lessons learned on previous versions of the guidances, to ensure that we also take into account your views. Then we had the public consultation where also you were able to provide additional information, and now then we ended with the adoption by the panel. I would like to thank all the colleagues, the speakers, the stakeholders, and last but not least also the experts from the panel, of course, and the different working groups on all the work and the preparation of this renewal guidance, and also the preparation and organization of this meeting. We hope it was useful to you, and we look forward to have further interactions with you in the future, because we believe they're really of mutual interest for you as well as to us as risk assessors. With this, I would like to close the meeting. Thank you again, and as indicated, slides and presentation will be made available on the website as well as the recording. Thank you very much to everybody, and have a nice day. Bye-bye.